All right, so welcome back to EBE for a little bit more photosynthesis. What we've got here is the reaction center that we talked about last time, this little grouping of photosynthetic pigments associated with a couple of chlorophyll A's at the reaction center, and then this huge grouping of accessory pigments that funnel energy down to the reaction center. Right, these antennal complexes, otherwise known as photosystems, are organized within plants in a particular organelle, and that's the chloroplast. Okay. Should have talked a little bit about chloroplasts in MCB, but in case you didn't, this organelle here is an unusual one in that it has three membranes. Trying to draw out this. All right, so here are my three membranes. There's an outer membrane, an inner membrane, and then this third membrane located here, which is called the thylakoid. Okay. As with other organelles you've talked about, I'm thinking namely the mitochondrion, the membranes themselves help to form compartments. The inner and the outer membrane are actually very close together, much like the nuclear envelope, so there's no real compartment between those. But we do have a compartment here, all of this stuff that we'll call the stroma. And then we have another compartment that's formed right inside that thylakoid membrane, which I'm going to call the lumen of the thylakoid. I joke in MCB that all of the organelles have different names for their lumens. I think that's silly. Let's just call it the lumen of the thylakoid and we'll all know what we're talking about. Now, within the thylakoid membrane, right, see all these things here are often called stacks of poker chips or pancakes because that's what they look like. These little stacks here are often called grana. Singular is granum. But this is how they would be organized within that chloroplast. If we're looking for the antennal complexes in the photosystems, they are located in the thylakoid membrane. Right? If we're looking for these photosystems, this is where they're located, in the thylakoid membrane of chloroplasts. Okay? All right, so we've done hopefully enough anatomy here that we can start into this process of photosynthesis. All right, we have the players. Let's go ahead and do this process now that we know who's involved in it. Okay? All right, as an aside, I do a lot of these, but I'm going to do another one right now. Cellular respiration and photosynthesis are two of the uglier biochemical processes that exist out there. They've been around for a tremendously long time, and they've gotten more and more complex over evolutionary time, I'm sure. People, when they start to teach these things, they try to come up with some gimmicks that make it easier for students to understand the process. I'm not sure that these gimmicks often do that. I think in some ways they make things a little more confusing. But for example, in cellular respiration, we talk about cellular respiration being divided up into glycolysis, the preparatory phase, the citric acid cycle, and electron transport and chemiosmosis. Well, in reality, that's all cellular respiration. People divided it into those chunks, much like you divide a book into chapters to make it a little easier to read. So this idea of breaking a complex biochemical reaction into subsets or pieces or chapters is an attempt to simplify. And photosynthesis is no different. here, there are two separate chapters that make up photosynthesis, just as there are chapters that make up cellular respiration. These were done to make things easier. Okay? Also, with these complex biochemical processes, another thing people have tried to do is come up with names that are descriptive about what's going on. So initially, the citric acid cycle was called the Krebs cycle. Then its name was changed to the citric acid cycle to make it easier to understand, and then it was changed to the tricarboxylic acid cycle. The same thing has happened with photosynthesis. We have these terms, and then over the years they've changed. I need to show you all of the terms because they're all used depending on how old the person is that you're talking to. And even your textbook by Brooker is in fact a botanist, Brooker's photosynthesis chapter is riddled with these different terms because he's trying to include all of them at the same time. Right, so initially the first two chapters of photosynthesis were called the light reactions and the dark reactions. Okay, very simple. And that was because of how Calvin, the biochemist, worked out these reactions. They had a piece of equipment that they used where they could turn the light off and on. And they found that when they turned the light off, a bunch of biochemical reactions ceased, and that meant that it was less complex to try to figure out what was going on because all these other things weren't going on at the same time. So they were able to easily work out what they call the dark reactions because these happened when the light was off. They called them the dark reactions. And then the other sets of reactions they knew kicked in when you turned the light back on. Hence the names, the light reactions and the dark reactions. Well, it didn't take folks long to figure out that the dark reactions were actually happening all the time. You just didn't see them when the light reactions were going on because they got swamped out by the amount of light reaction happening. So the names were then changed to the light-dependent reactions, which is the same thing as the light reactions, and then the dark reactions were renamed the light-independent reactions. A set of reactions that light doesn't matter, and a set of reactions where light does matter. Well, they decided these weren't very descriptive, so they came up with another set of terms. The first set of reactions were called the energy transduction reactions. And the second set of reactions were referred to as the carbon fixation reactions. And this set of reactions is also called the Calvin cycle because Calvin is the one that worked them out. So all of these are synonyms and all of those are synonyms for the same thing. I'm going to try to use these. These are the ones currently used today. The energy transduction reactions and the carbon fixation reactions. But I will slip. There's no way around it. I've had exposure to all of these, so it's just a big jumble in my head. What let's do now is let's just talk about these two sets of reactions in a very general way, kind of like a Sparks Notes or Cliff Notes approach here. So these are the energy transduction reactions. And here are the carbon fixation reactions. These are all part of the same process called photosynthesis. It's just we've divided photosynthesis into two chapters. A set of reactions involving energy transduction. In energy transduction, what you do is you start out with energy in one form and convert it to energy in another form. So in the energy transduction reactions, we're going to take light and we're going to convert that light into ATP, okay? And we're going to take electrons from water and we're going to move these electrons to an electron carrier called NADP plus to make a reduced form called NADPH. So NADP plus 2 electrons and 2 hydrogen ions gives us NADPH plus a hydrogen ion. Okay? That's the oxidized form of the electron carrier. That's the reduced form. It's very similar to one you've seen in cell respiration called NADP. NAD plus giving you NADH. This just has an extra phosphate group. It's a electron carrier used in the chloroplast. Okay? So I'm converting light energy to make ATP, and I'm taking electrons away from water. I'm splitting water, and I'm moving those electrons to NADPH, the reduced form of the electron carrier. So you could say, really, that the two main functions of the light reactions are to manufacture ATP and NADPH. Right? I want to manufacture these two molecules. That's the main job here of the light reactions, the energy transduction reactions. Okay? Convert light to chemical bond energy, reduce NADP plus to make NADPH. Okay? These reactions occur at the thylakoid membrane. That's where they occur. Okay, so in a nutshell, this is what we're going to be doing here in a little bit, is the energy transduction reactions to manufacture these two molecules. Okay. Number 2, carbon fixation reactions. In the carbon fixation reactions, I'm going to use ATP and NADPH. And I'm going to use those in addition to carbon dioxide to make carbon-carbon bonds. Specifically, this molecule is what I'm going to make, that triose. So I'm going to use the ATP and NADPH I made in the first part of photosynthesis. I'm going to take carbon in the form of CO2 from the atmosphere and use that carbon to make triose, and all of this is going to happen in the stroma. These reactions are using the ATP and NADPH made in the light reactions, and these reactions will continue until the point where the ATP and NADPH have been consumed. So when Calvin turned the light off in his chamber and worked out the Calvin cycle, he basically was able to follow these reactions for about 20 minutes, because that's about how long it took the ATP and NADPH to be used up, and then the carbon fixation reaction stopped for the lane 2. Okay? All right, so in a nutshell, these are the kind of the important components that we need to look for as we go ahead and do this process in more detail. I have this nice little slide which is trying to show us exactly what I just talked about. The light energy being converted to ATP and NADPH, water being split to release electrons, and then the Calvin cycle, otherwise known as the carbon fixation reactions, uses ATP and NADPH and CO2 to make triose. And that, in a nutshell, is photosynthesis. Okay? Now what let's do is let's go into this in a little bit more detail. Okay? All right. Here we are at the thylakoid membrane with the lumen of the thylakoid right here. And then out here, this is the stroma. Okay? So we're in the chloroplast looking at a portion of the thylakoid membrane, and this is the stroma, and that's the thylakoid lumen. Now, here we have some photosystems. The first photosystem that we have, interestingly enough, is called Photosystem II, because the photosystems were discovered in the wrong order, and nobody ever fixed it. So we're going to start this process off actually with Photosystem II. So Photosystem II is sitting in the membrane, and we're in the energy transduction reactions, the light reactions. So light energy strikes this photosystem. We get resonance energy transfer until we get to those two chlorophyll A molecules in the center of the photosystem II. The energy is passed to them, and then that energy is used to elevate those electrons away from the magnesium in the chlorophyll A. Now, once these electrons get far enough away, they get grabbed by an electron carrier. This is called plastiquinone. I don't need you to know the names of them, but PQ stands for plastiquinone. And then the next protein in the chain grabs the electrons. This is the cytochrome B oxidase protein. And then once it grabs the electrons, plastocyanin grabs the electrons. And then And then once plastocyanin has those electrons, we would essentially halt the chain right there. Unless we had somebody to take electrons from plastocyanin, in which case we could keep the electrons moving through the chain. So to do that, we'll put another photosystem in here. This one's Photosystem I. Light strikes it, elevates the electrons. And then these electrons are grabbed by FD. And then finally, NADP plus reductase. Okay? So then the electrons would go to here. NADP plus reductase